Welcome back to the Human Life Expectancy channel. I'm your host, Tom Nodine. This channel focuses on technologies that offer strong prospects for increasing lifespan and health span going forward. One of the most promising technologies is epigenetic testing, which uses blood or saliva samples to tell you your biological age, which can be either younger or older than your chronological age. It tells you what diseases you have or have had, what behaviors and environmental factors might actually be increasing or reducing your lifespan. So we keep a close watch on recent studies and advances in epigenetic testing. Since the first biological clocks came out in 2011 to 2013 at prices of about $2,000, there are now multiple scientifically validated biological clocks that cost between $200 and $300. The keys to refining these predictors further are conducting large volumes of tests and also further reducing the costs of the tests. The large volume, of course, gives us more data on more people over longer periods of time so that we can get more and better predictors. And of course, the lower the price is, the more they can be commercially adopted, which then helps us to get the large volume, then we get this virtuous circle going. So with all that in mind, my jaw dropped last year when I ran across a study from a group of scientists from Harvard Medical School who found a new way of processing epigenetic tests that they call time-seek. And it reduced the cost of processing epigenetic tests up to 100-fold, down to, in the extreme, $1.50 per test, while benchmarking favorably against current methods. And this is of obvious importance to us at Human Life Expectancy, Inc., insurance companies, and other companies that are in the process of commercializing these tests. It's also critical for advancing the science of epigenetics because these lower prices greatly increase the number of tests performed, expanding the data used to produce and to refine predictors exponentially. So, last fall at the Biomarkers of Aging conference at Harvard Medical School, I met one of the authors of this study, Dr. Jesse Poganik, instructor in medicine at Harvard Medical School. I'm delighted that Jesse is with us today to discuss these important studies. And just as a note, I'll put references to all these studies in the notes section of this video, and they are absolutely worth a close look. Jesse, welcome. How are you?
Good, thank you. Pleasure to be here.
Great. Well, let's start right off. Um, you know, let's start with the time-seek study, which you wrote, I believe, with Patrick Griffin, Vadim Gladyshev, and David Sinclair, and others. What is time-seek, and how did it achieve its results?
Yeah. So first of all, just to highlight, this was primarily the work of Patrick Griffin, who at the time was a PhD student in David Sinclair's lab, and we played a small role in helping the study out. But nevertheless, so what Patrick did was that he recognized this issue that you highlighted in the beginning, that there's this inherent problem with the cost of these tests to being able to scale them. And we really do need to scale for reasons we can talk about as we go on. But the basic idea that Patrick had was, he said, when we do these epigenetic tests, typically we're testing thousands or millions of CPG sites, not all of which actually go to the calculation of the clocks. So for instance, the very famous Horvath 2013 clock, that has only 353 sites that are used to generate the prediction of biological age. So Patrick's idea was, why don't we only take the sites that are actually used in the calculation of the clocks, only sequence those, and cut out all of the extraneous information. And so Patrick used a sequencing-based approach, which integrated this enrichment step, which selected only the sites that he was interested in profiling.
Mm-hmm.
And the nature of sequencing, really, just from a kind of general practical perspective, is that you pay for what you generate, right? So it means that there's kind of a fixed cost for the amount of data that you can generate on a given instrument, given technology. So you can maximize the value by sequencing only the relevant information to your study. And that's really the trick, if you will, with time-seek, that you only sequence the relevant sites, and you don't sequence the other things that are not relevant to your study. And by doing this, what it means is that you can, in a single sequencing run, you can add more samples than you otherwise would be able to and you ultimately drive the per sample cost down. So that's the whole trick of TimeSeek.
Okay. And if I hear you correctly, that part of that trick also is the multiplexing. Because you focused in so narrowly on the set of CPGs that you really focused on, it allows you to add data, similar data from many different people into the same sequencing algorithm such that you can do much more. And that's what the multiplexing part is. Is that right?
Yeah, that's basically right. So the idea is that, in the sequencing reaction, it's basically just it's just a chemical reaction. You add one base at a time. The machine can read what that base is based on various dyes. We don't need to get into the technical nitty gritty, but the point is that the sequencing is just a chemical reaction. There are four base pairs in DNA. You add one at a time. That's it. So the trick is, of course, you dump in a ton of samples, right, from different people, and you generate your sequencing data. But at the end, you need to be able to separate it so that you say this read belongs to patient one, this read belongs to patient two, on and on and on. So yeah, the idea of multiplexing is just that one patient gets one barcode, as they're called. This is just kind of a unique DNA sequence. You read that DNA sequence, and then computationally you can say, this barcode corresponds to patient one. I put that read into the patient one bin. This barcode corresponds to patient two. I put that read in the patient two. You repeat this, on and on for as many patients or as many different samples as you have in your data set, and then this allows you to deconvolute the data and assign the reads to the individual people.
Yeah. So my science director explained it to me as kind of like license plates on a car.
Mhm.
You tag each bit of data with a license plate or the specific source from which it comes,
Yeah,
and that
that's a
allows you
good
to
analogy.
it allows you to do many in parallel.
Mhm.
And so reading into this though, doesn't that presuppose that you're pretty darn sure exactly which CPGs you're looking for, and that therefore this is more appropriate for, I'll say, descriptive analysis than exploratory analysis, where you're looking at a large range of CPGs?
Yeah, yeah, that's one way to think of it. I mean, it's certainly true that you are, in some sense, more limited. When we do kind of the most common competing technology, which are these methylation microarrays, the latest version of those are measuring, somewhere around 1 million CPG sites for a single sample. So, you can look at that on the one hand as saying that's, several 100,000 more than I need to calculate my clock. On the other hand, if you want to do discovery, if you want to make new biomarkers, if you want to refine your biomarkers, you want to do deeper epigenetic insights, things like that, then there's an argument for generating the data. But the trade-off, of course, is that the price is much higher.
Yeah.
So it's we often have this problem, I would say, in science between the practical and the ideal, right? And it really ultimately depends on the purpose of your study, because both approaches, I would say, have validity. But it
Yeah.
is true that there is a sense in which a time-seq or a targeted sequencing approach is somewhat limiting. But if you're very confident about your biomarker, or at least about the fact that there is a particular biomarker that you want to assess in your analysis, then I think it makes sense
Hmm.
to go for a targeted approach.
Yeah. But I think you reveal important background here, which is that there the processing technology is kind of in the process of changing. For the past 10 plus years, it really seems like arrays have been the way to go. Prepackaged arrays from companies like Illumina, the Epic chip, and others that do a standard query of a broad range of CPGs. And it seems as though the costs of sequencing are coming down all the time, too. Is that the way you understand it?
Yeah, so from my perspective, speaking as an academic researcher and just talking about what we're paying in the lab for these analyses, the price of the arrays has remained pretty steady. Um, and in fact, even gone up as the number of sites that the latest generations can profile increase.
Hmm.
Um, on the other hand, the reality is that sequencing is becoming cheaper all the time. So, about, I would say 2 or 3 years ago, don't remember the exact date, but there was from my perspective, this real watershed moment when Ultima Genomics, another sequencing company,
Yeah.
announced this $100 genome. And that
Yeah.
sort of really kicked off this race among these providers to get cheaper. Indeed, like we do a lot of RNA sequencing in the lab. And these days, for a complete transcriptome, we're paying, right around $100. And that's pretty incredible for the volume of data that you get. And it seems to be the case, and I think there are good reasons to believe this, that it's going to continue going down. Um, the arrays, it's not clear that they would be able to go down just because of the nature of the technology. You can reduce the cost, and there are efforts to reduce the cost by reducing the number of sites.
Mm-hmm.
But again, you go back to this question about the trade-off between the breadth of your data and the specificity of what you have. It's not
Yeah.
to say one is better or worse, but I do think it's true that uh, the trajectory seems to be that the sequencing is getting cheaper all the time. The arrays are kind of stuck.
Yep. We see the same thing. We see the same thing. So fundamentally, if you look at factors that are driving costs of epigenetic tests, there's the processing technology itself, as you and I are discussing, the sequencing versus arrays. Arrays have been very predictable and easy to work with, but perhaps inherently unscalable. Whereas sequencing, again, much more customizable to a specific set of CPGs that you might be looking at, but much more scalable. And then so the second factor is scope, right? The breadth of the CPGs you're querying. The broader the scope, the more expensive it's going to be. The more narrow it can be, the less expensive it can be. And then we talked about multiplexing as being a really smart way that TimeSeq uses to put the data from many different people into one data sample to make it much less expensive for the processing. And then I suppose there's even a fourth factor which we haven't discussed, but I think it's pretty self-evident, which is that the more testing you do, the higher the volume of tests, generally the lower the cost gets, just as you get better and better at processing and that sort of thing. So all of this leads to the notion that the costs are going to be going down with TimeSeq. I was pleased also to see, though, that these reductions in cost came while at simultaneously benchmarking favorably to traditional approaches. Were there any tricks to bringing that about?
Beyond what we've discussed, I don't think so, because what has been known since, as you mentioned at the beginning, 2011, 2013, is that you can make these robust biomarkers from a few hundred, or even down to something like 7 CPGs.
Mm-hmm.
Which is not to say that that there are not value in the there are not different values in the clocks that use more sites. But anyway, the point is that it is. I would say it's not terribly surprising that you can build a clock, using few hundred CPGs, using this kind of technology. In fact, one interesting observation that many people in this space have made is that the clock sites themselves are not that special. So you can do this trick where it's not really a trick, but it's an observation that many people have made, which is that
Mm-hmm.
you can have a good biomarker in your epigenetic data. Say it's made of 500 CPG sites. You can remove those 500 CPG sites from the data set and build almost an equally, if not comparably good biomarker from the remaining data. So the nature of the sites themselves
Mm-hmm.
are not necessarily special.
Well,
They're
hold on.
reason.
I mean, is that because the markers themselves, no markers are particularly good,
No,
or is it?
yeah, I don't think so. I think it's really more about the way that these biomarkers are built, and that's typically using penalized regression approaches, which
Mm-hmm.
means that if you have two sites that are correlated with each other, the algorithm's going to throw them out.
Okay.
Because what you try to do with these approaches, generally speaking, is to select the minimum number of sites that explain whatever you're trying to explain in the data, whether that's chronological age, mortality risk, whatever it is that you're trying to predict.
Mm-hmm.
So it's part of the nature, or even an artifact, if you like, of the of the construction methods, the methods that underlie the construction of these biomarkers. And we
Hmm.
should probably just take a step back to say, I'm sure your audience is familiar with this, but it probably bears a little bit of emphasis.
Please.
When we say biomarkers in this context, what we're really talking about are algorithms
Hmm.
that integrate data from a few hundred or a few thousand CPG sites in a particular mathematical equation to produce a number that is representative of, again, whatever you're trying to predict. That may be biological
Hmm.
age or mortality risk, whatever. But I point this out just because this is not the conventional meaning of biomarker. But when we say biomarker of aging, this is what we're typically talking about.
Great. Thank you for the clarification. And so, I mean, let me ask, I mean, it sounds like it may actually be a function of the robustness of the underlying biology that
Mhm.
you can get similar results with a range of different CPGs. Is that a fair conclusion?
Yeah, I think so, and we can talk about that. I think when we get into the integrative epigenetic transcriptomic paper, because that was part of the motivation behind that study. But just backtracking for a second, because I don't
Hmm.
think I actually answered your last question after my tangent. But so what Patrick did was that he, his intention, I would say, in that paper was not necessarily to retrofit the existing clocks, for instance, the Horvath clock, a Grimmage, or something like that, to the time-seq method. He was building, a de novo clock essentially from his time-seq data. So he selected, sites that he selected. He did the sequencing. He built clocks. They were robust clocks. So when we
Hmm.
say that the performance was on par to the existing clocks, in terms of metrics like correlation with chronological age and association with some endpoints that he evaluated, those kind of summary statistics were what's on par with existing clocks. But um, the time-seq technology has not been specifically adapted, at least not that I know of. And Patrick has since moved on to a postdoctoral position at the Broad Institute, doing a lot of other cool stuff, but I think not time-seq so much anymore.
Yeah.
Um, but
Well, I'd,
anyway,
I'd asked you about this when we first met because that's kind of a core thought I had, which
yeah,
is, well, number one, this is fantastic, right? My goodness, if you can reduce costs by up to 100-fold, that's extraordinary in terms of its implications for commercialization. But yet, I think I'm noticing that the world, it hasn't rocked the world per se. Not everybody is doing time-seq and only time-seq at this point. And so you had mentioned that there may be some academic incentives that are a little bit different than commercial incentives, and also Patrick just moving on to other things. But I was surprised about that. I mean, are there any other kind of reasons you can think of for why this hasn't caught on in as the blockbuster that at least I understand it to be?
well, I think one thing that comes to mind is that it takes a lot, I think, to convince a commercial sequencing provider to onboard a new assay, especially when they would have to build it from the ground up, which presumably is what would have to happen here.
Mm-hmm.
And I'm saying in a hypothetical situation where we would say to a provider, we want to replicate, for instance, Grimmage, but with a time-seq approach. So
Mm-hmm.
on the one hand, you'd have kind of the R&D part of that. You need to design the probes to do the enrichment for the Grimmage sites. You'd have to check that the Grimmage sites that are captured by a time-seq like approach would be comparable to what you get from the arrays. You would ideally want to take some of the same samples as a technical calibration to make sure that you get the same answers between
Mm-hmm.
time-seq and arrays. Another thing that we haven't really touched on yet is this idea of coverage in sequencing.
Yes.
So in sequencing, of course, you need to have a specific number of essentially times that you read the same sequence to have a reasonable estimate of, in the case of methylation, the fractional methylation.
Mm-hmm.
And the estimate for that to make it on par to the array is actually quite high. So it's not a straightforward comparison, but it's been estimated by some of my colleagues to be on the order of 100x or more, so 100 reads of the same site,
Mm-hmm.
um, to get you onto the sensitivity of the arrays. So all of that technical lift would have to be done to establish the assay. And then if we talk about scale and the practical implications, then you'd have to convince a lab or a company, whatever, to onboard an automated processing pipeline for this
Yep.
assay, and that's
Yep.
a big ask, right? So
Hmm.
it's one thing if a company is, and I'm not speaking as a sequencing company, but right, just
Of
my
course.
experience based on working with many of them over the years.
Hmm.
But it's one thing if you're a sequencing company and you're onboarding, say, an RNA sequencing pipeline, right? Because every lab in the world essentially is doing RNA sequencing, so there's going to be clear return on investment, or probable return
Hmm.
on investment, right? But something that may be considered more niche for various reasons, like time-seq, that might be a bigger ask for a company, unless you can
Yeah.
kind of guarantee that the volume is going to be there. So that's kind of on the practical side, and then more on the just like the why,
Hmm.
that part of the question. I mean, that one's a little harder to read the tea leaves. One thing I think is that you know um, while there have been a lot of efforts towards validation of these biomarkers, and many of them are validated to some degree, there is still pretty long road to go. We think a lot about these biomarkers in the context of clinical trials, which means FDA approval, right? And so there's a huge benchmark that needs to be, or there's a huge target that needs to be met, burden of proof that needs to be met to make that
Yep.
happen. So, you know, I think that the way that some people think about this is just that we have the clocks. They were based on this technology. We have the technology available off the shelf, and we really want to progress towards validation. Let's just use the existing arrays.
Yep. Got
So,
it.
and
Got it.
then, yeah, if I could just throw in one last thing, and then I'll stop.
Sure. No, no,
we
please
you know
go. Please go.
there's also weird technical stuff that I think is worth saying. So, like,
Okay.
uh, over the course of the last however many years it's been, Illumina has transitioned from, just to give an example, Illumina has transitioned from the 450K human array to Epic V1 to Epic V2 now,
Hmm.
and in principle, so in each iteration they've added some probes, but in principle, the previous probes were carried through the different versions. Nevertheless, we've run samples on the same samples on these different arrays and gotten essentially different results. It's not to say that, yeah, it's not to say that, like, in one study something was predicted as 0 years old, the other studies predicted it as 100 years old. But what
Hmm.
we've seen are these weird technical effects where, for instance, you have a distribution of your data in the first array, and then you have like a systematic offset very slightly in one direction. So the data are still reasonable in the sense that you have a population over age. You can show correlation with age. The older samples are older, the younger samples are younger. That makes sense. But you can introduce these weird little technical artifacts when you change
Hmm.
technology platforms. And I think that's another barrier to being able to do it, because people don't necessarily want to have to deal with that headache, essentially,
Yeah.
until we're forced to by Illumina saying that they no longer make the previous version of the arrays, or
Yeah.
something like that. But the point is that there's a very large kind of chasm that needs to be bridged on multiple fronts, I would say, to get us to this future where we're going to be doing sequencing rather than the older fashioned technologies.
That makes sense. So going forward, it's not hard to envision that an Illumina or someone like them might say, okay, well, we've had, the 450 chip, we've had the version 1, version 2 Epic arrays. Maybe there's going to be others, right? It's not hard to imagine. And in principle, those could be more focused ones rather than broader ones,
Yeah,
which could
they're
then,
doing it, Tom. They're doing it, Tom. So
Mhm.
the latest one the latest technology that they've announced in their array catalog is called the MSA, the methylation screening array. And so this
Yes.
is a more focused assay, where you can essentially load more samples per chip, but you get fewer CPGs per sample.
Yep. Yep. So technology is going to continue to evolve, right? But I do think what we talked about earlier is probably a good prediction for the future, which is that, at least our observation is like with yours, that sequencing seems to be gaining momentum as arrays are losing momentum right now. Well, let
Yeah,
me ask
we'll
you. Oh,
see.
go ahead.
No,
We'll
I
see.
was just gonna say, I mean, I
Yeah.
don't I don't know if they're necessarily losing momentum, because again, people people are still clinging, if you will, for various
Hmm.
reasons. So there may be maybe kind of a steady period for the arrays, but I hope that the sequencing approaches are going to become more popular as time goes on. I think it's
Yeah.
probably just a matter of time.
Yeah. Um, as fantastic as the results are from TimeSeq, let me ask, could they be improved from what you've achieved? Or is this like the gold standard?
Well, it depends what you mean by improved. I mean, if we, I think if a logical next step, if someone would be willing to do it, would be to kind of retool the time-seek approach to the most robust, based on the current data, epigenetic clocks that are currently assessed using arrays. So I'm
Hmm.
thinking of things like Grimage, Dunedin Pace. So if those sites could be adapted, the sites that go into those algorithms, if those could be adapted to a time-seek like approach, and the appropriate analyses be done to show that the performance is on par to what you get from the arrays, I think that would be very powerful, because that would then kind of give credence to this idea that we could potentially move forward, uh, in terms of scaling our efforts and applying these biomarkers to larger cohorts of people
Yeah.
than we can currently do with the arrays.
Yep. And speaking of better approaches, you had kindly shared with me a pre-published version of a study called Integrative Epigenetics and Transcriptomics Identifying Aging Genes in Human Blood. Very cool study. I appreciate you sharing it. Can I ask you a few questions about that now?
Yeah, yeah.
So, if I understand correctly, this study advances aging biology by finding a way to combine epigenetic and transcriptomic data to produce even better predictors from epigenetic tests. And so let me just get the basics out here. Who did you work with on this study?
Mhm. So this was the main idea for this was really spearheaded by Madi Mokri, who's a close colleague of mine, and we also worked with Kira Herzog, Ki Jun Ying, Vadim Gladyshev, many others. And this was done actually in the broader context of the Biomarkers of Aging Consortium, which you mentioned at the beginning. Um, so, we're a large group of people working together to try to ultimately advance these biomarkers of aging. And this paper kind of sprung from some of our efforts within the consortium.
That's fantastic, and I really loved the paper. Um, can you tell us just as simply as possible, although I know it's somewhat technical, what is it that's good and bad about current epigenetic tests, and how might transcriptomic data be used to improve them?
Yeah. So I think it's actually probably not that technical, and most people in the field, I think, probably would agree with the following. The advantage of epigenetics, or the main advantage of epigenetics, is that it translates well across cohorts. So, if you profile a cohort in the US and a cohort in Europe, cohort in Asia, you get similar results if you apply biomarkers that are based on epigenetics. Or even if you look at the behavior, for instance, of specific CPG sites across
Hmm.
populations, across aging. So the translatability is very good. The main limitation, is the biological inference that you can make from epigenetic data.
Yep.
And that stems from the fact that we really, at the moment, still do not have a good understanding of how epigenetic regulation controls downstream biology. And by downstream biology, what I mean is, for instance, gene expression, levels of proteins, levels of metabolites, things that we actually understand,
Yeah.
to kind of make the whole picture of biology going on. So we have this disconnect between what we can observe at the level of DNA methylation and what happens downstream.
Yep.
Now, for transcriptomics, the advantage is that it's much more straightforward to interpret what the level of a gene means, right? Because gene express there are complications also between the level of genes and the level of proteins, etc. But regardless, it is still much more straightforward to interpret what the level of gene expression means. The limitation with transcriptomics, and these, I should say, though, they have been less widely utilized in cohort studies than epigenetic biomarkers. But the difficulty has been that the translatability between different populations, different cohorts, different studies, even different sequencing batches tends to be higher. So this is a technical problem that has to be dealt with. But I think this has really been, one of the key impediments to developing robust transcriptomic biomarkers of aging. And I just want to quickly say there's another preprint that maybe we can put in the description
Yeah.
from my colleague Alex Tishkovsky, who's built really robust, and I would say probably the first example of a really robust transcriptomic aging clock
Interesting.
that shows really good repeatability across different data sets and different situations.
Great. So hold on. Let me take you back to make sure I'm catching this. So, if I understand correctly, on the epigenetics side, CPGs such as, let me just choose one, eLovel2, are ones that are strongly correlated with aging, but they don't necessarily predict changes in the expression of their associated genes. Is that right?
Well, yeah, that is what we found in the study actually, but this was something that one might not have necessarily predicted at the outset. So there's a general what you learn in college level molecular biology is that methylation within gene promoters tends to repress transcription, and demethylation tends to turn on transcription. But again, it's difficult because of a lot of technical reasons. One of them is that you have many CPG sites associated with a gene. Some of them are in the promoter, some of them are in the UTRs, some of them are in the intronic regions, and it's unclear how each of these contributes to the expression of a gene. And then you also have like these long range epigenetic effects, where an epigenetic site somewhere in the genome can affect the expression of a gene, a long way away. So
Yeah.
that's the difficulty in connecting the epigenetics to the transcriptomics. And this is not exclusive to aging biology. This is just our general state of knowledge of molecular biology at this point. But so anyway, what we did in the study to connect to your question though was that we gathered multiple large data sets from human cohort studies, as many large data sets as we could. We harmonized them so that we could try to combine them between the different studies. And at the level of DNA methylation, we found the sites that are kind of known already to be strongly associated with aging. eLovel2 is probably the most famous example.
Yep.
And indeed, it was the top CPG associated with chronological age in our study across these different cohorts. But this is not surprising because this has been reported many times.
Yep.
So the surprising thing was when we did the same thing for transcriptomic data, and we looked at the level of or we looked at the change in gene expression for the genes that are associated with these top changing CPGs over age. In general, we found very little overlap. So in other words, eLovel2, the methylation changes as a function of age. In the corresponding gene, there is no change. And for
Yeah.
eLovel2 specifically, there's no change because it's silent in blood. And I should say this study was in blood. So that's very interesting because it tells you that the biomarkers that are being built on methylation, while they are obviously faithful, surrogates of chronological age, obviously linked to important aging outcomes like mortality risk and disease, um, the connections, how these work through the actual biology, may be more roundabout than we might have expected at the beginning.
So in layman's terms, just because eLovel2 or another CPG is highly predictive and very useful for producing a biological clock, doesn't mean that it ultimately manifests itself in a biological change that's measurable,
Yeah, or
that
at
may
least
not be
one.
expressed.
Yeah, or at least one that we don't understand the implication of,
Okay.
in terms of the broader picture. And it probably bears saying that our primary goal, in the lab, is that we want to understand the biology of aging, and that's
Sure.
why we care about this type of thing. So it's not to discount the value of the epigenetic biomarkers. People fairly, I would say, criticize the so-called black box nature of
Yeah.
epigenetic clocks. On the one hand, as a biologist, of course, I sympathize because we want to understand the mechanisms, we want to understand the deeper biology that underlies these things. But on
Hmm.
the other hand, I don't agree with the conclusion that some people go to from that frustration, which is, well, therefore there is no value in epigenetic biomarkers at all because we can't understand them. Because
Yeah.
and I think it's just ridiculous, frankly, that people would think this because, uh, I mean, Tom, that the associations with things like mortality risk are extremely robust for these
Yes.
biomarkers. So I think they're clearly remarkable tools, and I look at it more as an opportunity to understand, the what is now a black box, but will someday probably be more well understood.
Hmm.
And so
But
this
I mean,
study is.
it sounds to me like this study, an objective of this study, is to shine some light on that black
That's
box.
exactly what I was going to say. Yeah.
Oh, I'm sorry, I
That's
interrupted you.
okay.
You should have said it. Yeah. No, I agree. And so the idea is using epigenetic and transcriptomic data together to identify multi-omic aging genes that could be used to improve predictors or be targeted by therapeutics to mitigate age-related declines and other important factors.
Exactly.
But so it's kind of taking the best from both sides. The cohort robustness of epigenetics, as well as the additional, I'll call it expression robustness, if that makes sense,
Yeah.
of transcriptomics.
Yeah. And the other interesting thing was that, well, among many interesting things that we found in that study, but one interesting thing was that for the so-called multi-omic aging genes that we identified, and very simply, these were just genes that had changes over chronological age at both the level of DNA methylation and the level of gene expression. So in the two different kinds of data that we had. If you looked at just the gene expression level in the different cohorts for those genes, that was much more replicatable than transcription as a whole. So it seems to be the case that those genes may be, if not more important, more kind of robustly changing as a function of age.
Yes. And so providing this, I'll use the word orthogonal view of a different way of looking at some of the same things offers greater explanatory power
Yep.
than it would on its own. Great. So anyway, just fantastic study on that. Is there anything else you want to highlight in that study?
Yeah, well, I think the reason that we were talking about this in the first place is that with the multi-omic genes that we identified in that study, we actually designed something that's, it's not exactly like TimeSeq because TimeSeq targets CPG sites, but we created a targeted sequencing assay for these multi-omic aging genes that instead of assaying specific sites, i.e. one position, it's sequencing regions that we identified. So these are regions with multiple CPG sites that are changing as a function of age. And Madi Mokri, in previous work of his, identified that there are these regions that robustly change. Specifically, they robustly gain methylation as a function of chronological age. And he showed that in some contexts these can be more informative than the single CPG sites. But at the end of the day, it's very similar to TimeSeq in the sense that we enrich for these sites, we sequence only those sites, and we kind of throw out all of the superfluous information. And that assay we estimate to be around $20 per sample. So we hopefully that'll see more use as we go along.
That's great. That's great. And I'd like to talk with you more about that because we may have an opportunity to use that very soon. Let's talk about this separately.
Sure.
Speaking also, though, of better predictors going forward, you, Madi, Steve Horvath, Vadim, also recently wrote a correspondence in Nature Aging called What Makes Biological Epigenetic Clocks Tick, which made 5 recommendations to potentially improve predictors of biological age. Can I ask you to explain what those 5 factors are a bit?
Yeah. So the let me just start by saying that it's widely acknowledged in the field, and I'm sure you know this, that these clocks are a composite of different signals.
Mm-hmm.
Um, So, for instance, one of the observations that people will often cite is that different clocks likely measure different aspects of aging. And that's, fundamentally because they are a composite of many different signals.
Yeah.
So what we did was we try, and I should say that this was a correspondence, but there's a lot of good experimental and modeling work that's trying to do the same thing, which is to essentially decompose the clock signals from this very complex ensemble of signals to the individual constituent parts.
Yep.
So we talked about 5 of these in the paper. So one of them was the difference between gain or loss of methylation as a function of age. One is the
Well,
mitotic. Okay, yeah,
actually,
we can go one at a time if you
let's
want.
go
Yeah,
one at a time because
sure, sure.
I think that otherwise they may go over a lot of our
Yeah, yeah,
heads
it's fine.
in the audience here. So the first one is separately examining DNA methylation gains versus losses.
Mhm.
So again, from a layman's perspective, it is not the case that things universally get more methylated with age or things universally get less methylated with age. What I believe we found is that some things get more and some things get less methylated with age, but both can be predictive of your biological age.
Exactly.
Have I got that right so far?
Yeah, it's exactly right. It's exactly right.
Okay.
So in reality, methylation is a chemical modification, and it's either there or it's not. So in some sense, the fact that we analyze fractional methylation, between 0 and 1,
Mm-hmm.
is a little bit strange, right? Because it should either be only 0 or only 1. But the fact is that we're analyzing, most commonly a blood or a saliva sample, which has a mixture of cells, which have different cell types. And so again, it goes back to this idea that what you read out is a composite. And one of the things that makes up this composite are the different cell types. And this is
Mm-hmm.
related to some of the things later. But anyway, the point is that you can have changes that reflect either gain or a loss in methylation. This can happen to different extents in different cell types, and yeah, it's important to measure both of them because the clocks themselves, at least at the point of calculating the result from a clock, they don't care. It's just
Mm-hmm.
you have a coefficient, you multiply the methylation value, you do this many times, you add up the result, you add the intercept, you get the answer, right?
Mm-hmm.
So again, it's just a mathematical equation. So the recommendations in this paper were kind of additional analyses that you should do if you're using epigenetic clocks. And
Yeah.
that first one was to separately analyze gain and loss because that can tell you, interesting facets of the biology. For instance, linking back to Mahdi's previous work, he found that gain of methylation as a function of age typically occurs at particular sites that are associated with this complex called PRC2.
Mm-hmm.
And we don't need to go into the weeds on that, but the point is just that you can find these aspects that were previously unknown if you do these simple things like just separate gain and loss.
Yeah. And to your point of decomposition, it may well be the case that the biology of them is driven by different factors, and by decomposing them that way, we'll be able to get more precise estimates of biological age.
Yeah.
So great. That's the first one, gains versus losses.
Yeah.
Let's go on to the second one.
Okay, second one is mitotic component. So mitosis, of course, is the process of cell division, as you know, and it is well known from studies in cell culture, for instance, that as cells divide over time, their methylation age increases, and there are specific sites that are associated with this. You can even build, and people have built, specific mitotic clocks which predict the number of cell divisions that a cell has undergone. So
Mm-hmm.
this is obviously an important component for something like, say, blood, where the turnover of cells is quite important to the biology of the system, and it's a signal that I would say clearly can be extracted from the overall composite again because the sites that change as a function of cell division are identified.
Mm-hmm. And I think the third one is kind of similar. This is controlling for cell composition.
Yep. Yeah, so this is a big one, especially with something like blood. So of course, blood is
Mm-hmm.
a mixture of many different cell types. We know now from a number of studies, especially those using single cell analysis, that different cell types, and particularly different blood cell types, age at different rates.
Yeah, like
So,
native and memory T cells, those
yep,
sorts of things.
yeah, exactly. So it means that if you have a shift in the proportion of your blood cells, you could get a different answer
Which we
in
know
terms.
happens with age, right?
Yeah, so this is the complexity. So
Mm-hmm.
here's my personal opinion. I don't know how much it reflects my co-authors. So I
Okay.
think that in terms of predicting biological age with epigenetic clocks, the cell composition component I think is almost like a red herring, because people sometimes throw it out there as a way to kind of discount the results.
Mm-hmm.
um, they say, "Oh, you just capture a shift in cell composition." But the reason I think that this is misguided is for the reason you just said, which is that it is known that cell composition changes as a function of age. And I'm a pretty simple-minded person. I like the if it looks like a duck and
All
acts
right.
like a duck, it's probably a duck. So from my perspective, if you have a shift in the cell composition that resembles the older state, you know, why is that not a reflection of an elevated biological age? So I don't think that there's anything special,
Mm-hmm.
or any kind of strict requirement that it must be an epigenetic change that is reflective of aging. I think the cell composition is actually a very important component of the signal. And actually, I think that in many clock papers, the original papers in which they were developed, they usually do so-called mediation analysis, where they try to remove the contribution of cell composition. And in many cases, the associations with, for instance, mortality attenuate. So it means that the composition is really important.
Yeah.
But anyway, going back to the paper, now that I've ranted for long enough, it's an important part of the analysis because it helps us to understand, the extent to which composition versus intrinsic methylation changes contribute to the overall signal. And I
Yeah, and
think
that should
that's
be
I think that's
discoverable, right?
important.
Right.
Exactly. Yeah.
It should be easy to, well, listen to me, but it should be relatively easy to decompose
Well, let me just
that
say
portion,
it
which
is,
is
yeah, it's becoming easy because there are now very good computational tools which, from the methylation data, you can very easily estimate cell proportions. In other words, you don't need to do a separate assay. You can infer these from the cell proportions, and if you want to, you can adjust your data, or you can analyze it as a component of your data.
very cool. Very cool. Okay, so again, a further decomposition in terms of cell composition as it changes over time. Two other factors, including exposure factors, I think is the fourth.
Mhm. Yeah, so I think this is important. This goes back to this idea of kind of intrinsic versus extrinsic aging. So on the one hand, you have the changes that happen or that are believed to happen just as a function of chronological age. But on the other hand, we know now from many studies, including studies that we've published, that a variety of factors in the environment affect your epigenetic and, we believe, biological age. So, for instance, we published a paper showing that very severe physiological stress can transiently accelerate epigenetic age. So, these can be important covariates in the analysis. If you have that information, or if it's an important part of what you're trying to study.
Mhm.
Another interesting example, just to give you, is that it's been shown in some cohort studies that generations that experienced famine have very
Yes.
different epigenetic aging profiles than
Yeah.
those who haven't. So yeah, the importance of the environment, I think, can't really be overstated. So it
Yeah.
should be. That's why we recommend that it should be included in the analysis.
And then the fifth one is kind of similar too. It's isolating other kind of exposure factors: smoking, drinking alcohol, UV radiation, etc. I mean, am I right on that?
Yeah, I think we skipped one though, which is
Oh,
the
okay. Go ahead.
differentiation and de-differentiation. So I would yeah,
Thank
I
you.
would group exposures with the environmental stuff.
Okay,
I
got
think
it.
that
Yep.
kind of all falls under the same umbrella. So the differentiation de-differentiation. It's good that we saved this for last because it's probably the weediest one. So this is just the idea that when cells differentiate, their epigenetic profile in general changes. So in fact, the epigenetic landscape is an important determinant of cell type.
Well, the Waddington plot, the Waddington
Exactly. Yeah,
field.
exactly.
Yeah.
So conversely, using the Waddington model, when you do dedifferentiation, which is associated with these epigenetic reprogramming technologies, you also change the basically the entire landscape of the epigenome.
Okay, so
So
this would be like using Yamanaka factors.
Yamanaka reprogramming, exactly.
Okay.
Yeah. Yeah. So it's an important component because you get beyond just the level of clocks, you get this really, drastic global reshaping of the methylome. And so that's why it's an important factor to account for in your analyses. And people, people also talk a lot about the difference between, particularly in the context of partial reprogramming, for instance, the difference between rejuvenation that is believed to occur under reprogramming and dedifferentiation, which is generally, at least if you push it too far, considered to be bad because that can lead to things like teratoma formation.
Yeah. To bring our listeners kind of back into this, Yamanaka, I believe it was 2007, discovered 4 factors that can be used to take a somatic cell and turn it back into its pluripotent state, which basically it reverses the whole Waddington landscape and says, whatever differentiated cell you happen to be now, boom, let's take you back to where you were before. And so, I mean, with that understanding, it's not at all surprising to me that this is a big factor. I mean, if you're trying to figure out the epigenetics of something, it would be wholly relevant to know whether it's undergone some sort of Yamanaka reversal to its pluripotent state. And that's what this is effectively saying. Is that right?
Yep.
Cool. So are you and other members of the teams currently working in these areas?
Well, I don't do a whole lot of reprogramming myself, but other people, a lot of people do. So there's a lot of exciting work going on there. My current efforts are, I would say, one of the focuses is on single cell approaches. So almost adapting what we've done in the integrative paper, but trying to do so at a single cell resolution. And the reason is because, a lot of the things we've talked about today, the idea of cell composition, the idea of differentiation, all of these things, the reason that we care about them in the context of epigenetic clocks that are being developed right now is because we are doing bulk profiling. We're taking a chunk of tissue and grinding it up, or we're taking a blood sample and we're sequencing the whole thing. And at the end, what you get, as we've said many times now, is a composite of different signals. And so there are a lot of questions out there that I think really require single cell resolution to answer unambiguously. And one simple example, I think, is the effect of blood cell composition. Right. So if we can sequence specifically different kinds of blood cells, and there's been some preliminary work out there doing this already, actually. But if we can sequence specifically different blood cells, we can know exactly which ones are changing in which ways, and how this ultimately contributes to to the bulk results we get. But this is just one example. There are many questions out there, I think, that are unresolved, that could really use answers and really need single cell approaches. So that's going on. We continue to work with the consortium to try to tackle the scaling problem that we've been talking about, which is that we really need more data. Essentially, is the easiest way to say it. And we need to apply these biomarkers in more different populations across the world to make sure that they are robust. And I think another exciting new avenue in the field, in the lab, but in the field more broadly, which we haven't touched on yet, is proteomic profiling. And this is very exciting because for about a year and a half now, there have been these very exciting preliminary reports in large cohort studies where people have built tools that can estimate biological age of different organs from a single blood plasma sample. So essentially, it gives you much more insight, across the different organs and the different systems than we can currently achieve with epigenetics. And so people are very excited about these. And the other exciting thing about those proteomic studies, just to say, is that they have, in some sense, they have achieved scale that epigenetic studies have not achieved. So on the one hand, I would say epigenetics have been, and DNA methylation clocks specifically, have been applied in more different situations, probably because they've had, now a 10+ year head start. But,
Hmm.
you can find data about your favorite disease, or, whatever the case may be. You can typically find methylation data out there. But what we don't have for methylation yet, but we do have now for proteomics, thanks to the UK Biobank, is a large study of essentially healthy people aging normally, with very, very detailed phenotypic data. So the UK Biobank has information on something like 7,000 different phenotypes, and they've now produced proteomic profiles for, I think, 30,000 individuals. And so the statistical power that you get from those kind of numbers is really transformative compared to what we
Yep.
have now for methylation. So I think that's really exciting for several reasons, and I hope, among other things, that it will demonstrate to people why we really, really need to scale, the methylation biomarkers, so that we can try to make orthogonal insights at the same level of impact that we're now getting from the proteomic biomarkers.
Completely agree, Jesse. I mean, that's fantastic. And it sounds like whether it's through decomposition, through the single cell work, which you're now doing, through scaling, through proteomics, or any of the other fantastic things going on in this area, there's a lot more room to learn more and to do better in this area. So delighted to connect with you on these. Thank you so much for bringing us up to date and for your ongoing research and insights. Let's definitely check in again in a few months to track this important area and see how it evolves in its ability to help us all live longer, healthier lives.
Okay, thank you for having me.